Hello, my name is Dana Gardner, and we're going to talk about the process of muscle contraction. So the first process that happens is we get a signal to our brain saying that we want to pick up a 20-pound dumbbell. And so that signal goes to our motor neuron, and these are our dendrites right here carrying the signal through our axon and then delivering to our muscle. And inside our muscle, we have our tiny muscle fibers containing our sarcomeres, which is our contractile properties. After the signal gets delivered into these sarcomeres, actually before that, the signal goes through to this motor end plate, and this is our synaptic cleft right here. And inside the motor end plate, it does contain synaptic vesicles containing our neurotransmitter acetylcholine, which is the key to muscle contraction. So after that signal reaches there, it releases the acetylcholine, letting it bind to its receptors right here along the muscle fiber. After the acetylcholine binds to the receptors, it then allows sodium to rush into the muscle cell, causing it to depolarize or making the muscle cell become more negative. After the muscle cell becomes more negative, it then signals an action potential, which goes down our T-tubule. And after the action potential goes down our T-tubule, it reaches our sarcoplasmic reticulum, causing calcium to flow out of the sarcoplasmic reticulum. After the calcium flows out of the sarcoplasmic reticulum, it then goes to our thin myofilaments, actin, and the actin contains our tropomyosin and troponin. Calcium binds to the troponin, which are these little red dots. After the calcium binds to the troponin, it allows the tropomyosin to change shape, letting the actin reveal its binding sites for the myosin. These myosin are thick myofilaments. So this is a close-up of our myosin and our actin, which is kind of going away. Since the myosin head is connected to the actin, it does have ADP and inorganic phosphate. And after that happens, ATP comes and binds to the myosin head, making it separate from the actin. So it releases, and then the ATP hydrolyzes back into ADP and inorganic phosphate and starts the process all over again, making the myosin head carry the actin together, causing the muscle to shorten, causing muscle contraction. And if the brain says they don't want to lift that weight anymore and they're done, there is no more ADP and inorganic phosphate to be hydrolyzed, so the muscle just relaxes.